All right, guys, we're going to pick back up in our notes with Roman numeral 6, functional systems. So what we've looked at pretty much so far are structural components of the brain, so actual structures of the brain. Now we're going to kind of pivot and we're going to look at when multiple structures work together for a particular function. And so this is what I've called functional systems. And there are many. I chose 3. So I chose the limbic system. I chose something called the reticular activating system, or your RAS, and then memory, the functional system of memory. So let's start with the limbic system. So if you know anything about the limbic system, you probably have it associated with emotion. So the primary function of the limbic system is to sort of perceive and process and relay emotion. There's also some memory involved in the limbic system because of a couple of structures. And so this might help you to kind of think about some memories provoke or evoke an emotion, right? The two are tied to each other. So the regions of the brain that are operating here in our limbic system is something called the hippocampus, which you can see as this green structure in this diagram right here. The hippocampus is also going to support some memory function, which is why emotion and memory are attached to each other. The other region in your limbic system is the amygdala, which is this little red circle right here. So the amygdala is the one that is the region that's more associated with emotion. And these are typically what are called survival emotions. So emotions like fear and anger, even emotions that are related to sexual behavior, the desire to reproduce, that is a survival emotion. And it comes from the amygdala. The third big region here in the limbic system is your olfactory cortex. So we have already seen that and labeled that when we talked about the sensory cortex tissue. So olfactory, remember, helps with smell. And so what the olfactory cortex is doing is information routed here that's going to help us to kind of determine what it is that we just smelled, which again is associated with memory and can oftentimes evoke emotion. The next functional system is called the reticular activating system. And so I actually want you to pause this video, right? So this is sort of like Scout's honor. Do your best here, pause the video, and I want you to go to Google Classroom and pull up the article that is titled RAS. It's a read with, I think, pretty quick read with 5 questions at the end. I want for you to go read the RAS article, answer the questions, and submit those on Google Classroom, and then come back to this lecture video and continue to take notes. I promise you that your understanding of the RAS will be much, much higher if you read the article first. So pause this video, pause your note taking, go read the article, and then come back. All right, so if you are back and you have read the article, you know that the reticular activating system is really working like a filter. So think about information, sensory information from all out in our body is coming towards our central nervous system. It goes up our spine, up our brainstem, and it passes through this reticular activating system. Some of that sensory information is allowed forward or allowed up even further to the thalamus, where it gets routed out into sensory cortex tissue. Some of that information does not need to be passed all the way up to your cortex for processing. Some of that information is not important. Background noises are a great example of things that really don't need to go up to our cortex for processing. So the reticular activating system will filter the sensory information, determine what's important, and pass the important signals up further to your cortex. Now, the RAS also helps with habit formation, which is something called habituation, because it lets us tune out these sort of everyday repeated stimuli, and we can focus on the habit, we can reinforce the habit, and form the habit as a result. The RAS is considered your brain's arousal system, which maybe makes sense if you connect it back to ADD and ADHD. So, your arousal system is either stimulating your brain a lot or not. And because it is your arousal system, it also helps regulate your sleep and wake cycle. So, we talked last week about this gland called the pineal gland that is producing melatonin. Melatonin helps regulate our sleep wake cycle. Melatonin acts on the RAS. When it is time to enter our sleep cycle, the RAS actually amps up its filtration and lets fewer signals pass up to the cortex so that our brain can kind of slow down and begin to turn off and we can go to sleep. When we are waking up, the RAS will reduce its filter and will start to allow more signals to pass forward and stimulate that cortex. There was a pretty significant connection in your article, and I hope that you saw, to ADD and ADHD. These are individuals whose RAS is really not filtering as much as it should. And so what's happening is that all these signals are being allowed to pass up to the cerebral cortex for processing. And that becomes really, really distracting. When you are taking in all of that sensory information, it's hard to focus on things. And so we see that as ADD and sometimes ADHD if we see the hyperactivity aspect of it. Now, taking stimulant medications, things like Adderall and Ritalin and Concerta, will actually stimulate the RAS to pick up its pace and to filter things better. And I like to point that out because oftentimes it's confusing. Why would we give someone who is already ADD or ADHD a stimulant? Because what we are trying to stimulate is the filter that will help to filter out some of that sensory information so that the individual can actually concentrate. So, structures of the brain, where is the RAS located? You can see it right here in the green structure. It mostly parallels the pons, which is this big bulge right here. There's a little bit down here in the medulla region, but your pons area is where your reticular activating system is located. Last system, I want to talk about memory for a second. So, forming memories is a bit of a process. And so I'll kind of talk you through the process and then we'll look at parts of the brain that are associated with the process. When you are trying to form a memory, you first have to take the memory in, and then you have to sort of remember it and remember it and remember it to make it stick. The longer you remember and remember and remember that memory, the greater the chance it gets stored in something called long-term memory as opposed to short-term memory. So there are three basic steps to memory formation. The first is called encoding. That's when you are taking in sensory information. That's when you are first forming the memory based on what you have experienced. From there, we roll into a period of what is called storage and consolidation. So this happens by sort of actively remembering the memory. And we can see in our diagram down here, once sensory information has come in, we're going to lose a little bit of it, but it gets passed forward into short-term memory. And if we review this, if we remember it enough, we move it forward into our long-term memory. A little bit of forgetting does occur as we are trying to shift from short-term to long-term. The third step is retrieval, and that's really what I was talking about, the remembering the memory, retrieving that information. from your stored memory helps it to stick even more. So areas of the brain that are involved in memory, the hippocampus, which we saw with our limbic system, this kind of interestingly enough is one of the first areas of the brain to be degraded in people that have Alzheimer's. And if you know anything about Alzheimer's, probably you know that the big symptom of Alzheimer's is losing memory, forgetfulness, and eventually losing memory. And that's because your hippocampus is degenerated. I'll come back to that video link because I do want to show it to you. The second area of the brain that is involved here in memory formation is the olfactory cortex. So again, smell helps us as a sensory input, helps us to form memories. Which is why you probably can smell something and you have a very distinct memory associated with it. Let's check out the hippocampus video. So this is kind of where we will end. This is pretty interesting. This video details an experiment that was done that really is what allowed us to discover the hippocampus's role in memory. So let's see this together.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube. But this wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used them to reduce seizures in psychotics, Scoville decided to remove HM's hippocampus, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM's seizures virtually disappeared, with no change in personality, and his IQ even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory, it redefined what memory even meant. One of Milner's findings shed light on the obvious fact that although HM couldn't form new memories, he still retained information long enough from moment to finish a sentence or find the bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM's mind could form the initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In a now famous experiment, She asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
Okay, so just kind of an interesting end there to this set of notes. So you are finished with this lecture. You are done, Zoe.